Okay, I think it's recording. Okay, hi you guys. Thank you for attending today's expert series talk. I hope you guys are excited for an amazing talk. So today we have Lauren, who is a marine biologist who is originally from St. Louis, Missouri, and is currently living in San Diego, where she works as an environmental consultant and as a biology lecturer for California State University of Long Beach. Her undergraduate degree was in biology at Florida State University, where she discovered her passion in fish and shark research. Lauren took that passion to an internship in the Bahamas, and then to the Pacific Coast, where she completed her master's at Cal State Long Beach with Dr. Lowe, in his lab in January 2025. So it's not that long ago. Yeah, and so like I'm really excited that you guys are get to hear her research because I thought it was pretty amazing. So yeah, I guess with that, I would leave it to Lauren.
Cool. Okay. Today we're going to actually head to the other side of the world. And we're going to be looking at site fidelity and movement patterns of fishes associated with Qatari oil platforms within the Arabian Gulf. So, oh, there it goes. So when we think about movement strategies of fish, two common, if we go very basic wise, we have site attached and highly migratory. So when we think about being side attached, some of the advantages of that are the resident advantage. So they have an advantage for competing for space. They can have better efficient resource acquisition when they're staying in the same place. They can also have better effective predator avoidance and knowing the place that they're staying. These are typically reef associated or benthic species. For highly migratory fish, some of the benefits for that, so being nomadic and moving around all the time, is they can move around to find better prey patches. They can also move around further distances for avoiding predators. They can also move around for more suitable environmental conditions. And so this typically includes large pelagic individuals. And so for our movement strategies of fishes, they're influenced by the capacity of environments need to meet these energetic and survival needs, either within a single location or across multiple locations within an area. And so the size of a home range of an individual can vary with the size of an individual, temperature within that environment, habitat composition and quality, maybe complex versus simple, how much food is available to them, how much shelter is available to them, and what kind of mates are available to them as well. When we think about complex habitats, home range size can be reduced within a structurally complex habitat. And that's due to the rugosity and vertical relief that contributes to this complexity, creating more food sources, more structure, including more homes for them. It can support also higher abundance and diversity. So when we add structural components, that increases more spaces for these individuals to occupy. When we think about offshore oil and gas platforms, these structures can provide a structurally complex habitat. They can sometimes give you shade. They have horizontal and vertical substratum that creates really great vertical relief. They also have tons of surfaces for encrusting invertebrates, which creates this really great rugosity underwater for these structures. There have been numerous studies around the world that have shown similar trends for these platforms being great environments. So Southern California platforms showed that 63% of tagged individuals remain there after 2 years. And then within the Arabian Gulf, platforms have shown some of the highest diversity in the region. So there are some things about offshore oil and gas platforms when they sign their lease. So they're only able to put them in if they're going to eventually take them out. So they must eventually be removed or decommissioned. Decommissioning of a platform is really, really expensive and can be dangerous for the environment as well as some of the people that are doing the decommissioning because it most of the time does require explosives. Rigs to Reefs came about to provide decommissioning options. So, for a platform to have reefing options, it must be established as an essential fish habitat. And what I mean by different decommissioning options is maybe different ways that we can actually leave these structures in place for these fish if they do seem to be a critical environment for them. So our focus was in the alkylate oil fields as they were considering initiatives for potential reefing. So that's going to be that red box that denoted on the graph within the Arabian Gulf. The Arabian Gulf has extreme environmental conditions, which makes it a really unique place to look at this phenomenon. So temperature ranges from 22°C, sometimes even below that in the winter, to 35°C, sometimes even 37°C in the summer. And so this happens on an annual cycle. And we also have salinity peaks sometimes of up to 70 parts per thousand, which is really comparable, or which is comparable to the Dead Sea. So really, really salty area. And the whole basin is very shallow. So we have only a max depth of 75 meters in some of those deeper areas of the Gulf, and which what this means is almost the entire Gulf is in the photic zone, so sunlight can almost penetrate the entirety of the body of water. Some of our research aims was we wanted to know if fish of two different ecotypes, whether are benthic or pelagic, so are nomadic versus more reef associated, would stay at platform sites for over an annual cycle. We hypothesized that residency time is going to vary between our benthic and pelagic individuals. And we wanted to also determine how fish are going to utilize this platform structure overall during extreme seasonal changes that occur every year. So our second hypothesis was that seasonal temperature differences is going to influence how these fish are utilizing depth on these platforms. And then finally, we wanted to look at the connectivity of platforms within the oil field. Are fish going to treat multiple platforms as one continuous habitat? And we did hypothesize that interplatform fish movement is going to occur frequently. So when we look at our study site and the alkylate oil field, our study sites are denoted in those red stars, but I did want to highlight that within this oil field, all of those colorful lines in between these platform sites are denoting epibenthic pipelines and outfall pipes. And so that means that they're above ground, so they're able to be visible on the bottom, connecting each of these structures to one another. We're specifically looking at just three platform sites of interest. They're within similar proximity to one another, maybe giving us a better idea of some of the traveling movement in between. I also do want to mention that DP2 and PP1, while we are treating as one site, is an interconjoined platform, so it's slightly bigger than DP1 and DP3. Our monitoring took place from the 31st of May in 2023 to the 1st of June in 2024. We included 2 acoustic receivers per site, making a total of 6 receivers, where we had receivers placed 10 meters below our catwalk on our platforms on opposing sides of the platforms, except for the case of DP2 and PP1, where they were just hanging from the center of both. So, what are these acoustic receivers and what are they doing? We were using acoustic telemetry to be able to track presence and absence in some of these depth movements that we might see on platforms. So we see 2 acoustic receivers hanging from that catwalk in this animation. And so what happens is these acoustic transmitters are going to be surgically emplaced in fish. And whenever that acoustic transmitter is within 300 to 500 meter range of these acoustic receivers, the acoustic receiver will actually pick up that ping sound and it will report to us the tag ID, the receiver that picked up that tag, the date, and the time in which the detection was picked up, and we can Bluetooth download that from the receivers. For depth, we get the same information, except we also get a depth associated with the fish of where it was at the time of the detection. So for tagging, majority of our fish were cut through hook and line, but we did catch 2 through a baited fish trap. There's going to be 50 individuals included in the final analysis where we surgically implanted those transmitters among the 3 platforms, and that encompasses 13 different species. 28 fish are going to be considered benthic or known to rest directly on the sea floor, and 22 fish were going to be considered pelagic, known to range more in the water column, more of our nomads. And 15 of these transmitters are pressure sensors for our depth readings. So this is just a tag table of some of our individuals, including some of the pictures and size ranges. We'll get more into that in a little bit. So jumping straight into our residency, or our hypothesis 1, looking at residency time in between these 2 ecotypes. So residency is just going to be denoted, our residency index is just total days detected over total days at liberty, or total days available to be detected within the study. So the number is just going to range from 0 to 1. A fish was going to be considered present if it was detected at least twice in one day. And this is going to be an indicator if habitat supports daily biological requirements for these individuals. So our first plot that we see here, we have residency on our y-axis, and then our x-axis is going to be our species split by benthic in orange and pelagic in blue. This is just a crossbar plot, and each point is going to denote the average residency time of our individuals throughout the study. Our ANOVA showed that we did have a significant difference in residency between our two fish types, between our benthic and pelagic. And when we break that down a little bit, so high residency, we're considering anything from 61 to 90% of days, or 61 to 100% of days spent at these platforms. We only had two pelagic individuals, a longfin batfish and one giant trevally considered to be highly residential. But we had 15 benthic individuals encompassing a few different species to be highly resident at these platforms. And this is also an accumulation of all three platform sites. When we look at moderate residency, anywhere between 31 to 60% days spent on any of these platforms, we had seven pelagic individuals and we had six benthic individuals. For our low residents, anywhere between 0 to 30% of days spent on platforms, we had 13 pelagics and we had 10 benthic individuals. So when we think about the explanation of that, we do see a large difference in our residency between our two fish types, where we have lower residency for our pelagics and higher residency for our benthics. And we also do have some variations, even just among species, between our residencies. So we saw variations in residency across and within our species, and this could likely reflect density-driven carrying capacity as one of the explanations. So these platforms have been in place since 1997. And so they're pretty well established. And so what we could be seeing maybe is a carrying capacity limit at each of the structures where they can only hold and resource so many individuals. Also, maybe some species specific behaviors when we look at two of our pelagic individuals that showed higher residency time. And we'll get in a little bit more about those individuals later. But the other thing too is maybe sample size limitations. So we did encompass 13 different species, but we only had 50 tags. And so we didn't have a really good even split of sample sizing a bunch of each species. It was basically a little bit of a lot of different species. And so that could also kind of contribute to this. When we think about the orange spotted trevally, the fish on the right that we see there, they're really economically valuable in the Arabian Gulf and kind of actually a lot of other places as well. But that high residency is a really positive thing that we can see because these platforms may provide refuge from coastal fishing pressure, but kind of still keep them in the economy, which is great. We did have some ROV footage from a collaborator that did ROV footage of these platforms specifically, and they showed that some of the batfish had really high abundance at our platform DP3, but also at one of our unmonitored platforms, DP4. And so this might explain some of the interspecific differences in RI. While some of them had really high residency and some of them had lower residency, they might actually just occupy multiple platforms at once. And when we kind of jump into looking at some of the differences between platform and season, I ran a two-factor ANOVA looking at this. We have a residency again on our y-axis. We're going to again have benthic in orange and our pelagic in our navy blue, and it's going to be split seasonally as well as by platform. All of those different shapes are just all those points are still just an individual, but all the different shapes are just denoting which species they are. You don't have to pay too close attention of that because we actually didn't have really any significant difference in residency among our three platforms. We also didn't have any significant differences in RI among seasons for either of our fish types, and that was what we ran a Cresco Wallace for. And so, what does that mean? It might just mean that all these platforms provide comparable habitat to one another. We were definitely kind of surprised with our interconjoined platform as it is larger, but maybe that's a good thing. So, some of our high RI to our high residency of our mature individuals across seasons could potentially indicate spawning. So, some of the mature individuals with high residency include all our tagged grouper, our two-bar sea bream, and our longfin batfish. And this is really great because if they're staying there throughout all seasons and they are spawning every year, that could actually increase productivity for these platforms, which is a really good sign for these environments. And we do see evidence of spawning behavior at platform DP3 that we actually caught on ROV footage. from some of these individuals, like our giant trevally. So far, this is really good indications that we're seeing as a positive habitat. And then we kind of wanted to also break it down by size. Is there any differences between whether you're older or younger by benthic or by ecotype? And so we ran a regression, or I ran a regression, where we have residency again on our y-axis, but this time we have standard length in centimeters on our x-axis, and this is just our pelagic individuals, so each point denoting a pelagic size. And we do see a nice actual decrease in residency as our pelagics get larger. So smaller pelagic individuals showed higher residency, and that was a significant correlation for us. And when we look at benthic individuals, they showed no difference in RI with size. Or again, we have the residency on the Y and standard length in centimeters on our x-axis. And so what does that mean? So Lowe and Bray's cost optimization model may explain this. So fish with lower affinities to structures, such as our pelagics, do tend to expand their range as they grow. But territorial fishes may exhibit the opposite trend as effective territory holders. So some of our epinephrous species, our grouper species, could be territorial. And so what this means is they actually increase space use, but they don't actually leave the site in which they're at. As epinephrous is one of we have a few different species of our groupers, and that does make up a good chunk of our benthic individuals. So that could explain some of those pattern differences that we're seeing. So for our main takeaways, we do see that platforms provide essential needs for some individuals with that high residency. We do have inter and intra-species differences, which could be correlated for a couple different reasons, including small sample size. Some individuals could be spawning and increasing the productivity of this habitat. And size is an important contributor to our pelagics, but not significant to our benthic residencies. So jumping right along, moving to our seasonal temperature differences will influence fish depth use on offshore platforms. So first, just to show you guys again, this is our depth tagged species included in analysis. So we have pelagic in our blue and then benthic in our yellow, where we have few different trevally species along with our longfin batfish and our pelagics, and then two different groupers with our mangrove red snapper and our spangled emperor. And we will kind of come back to these also in a little bit. So when we look at our overall depth utilization by fish type, we just ran a t-test looking at what kind of depth use was going to be different between our two types. And when we first look at our benthic fish, they utilize deeper areas of the platform more than not, but they also did have a pretty wide range in the depth use that they had. But there was no dial differences, so there was no differences between our day and our night detections. So also I should probably break this graph down to you where we have total depth on our y-axis, but it is inverted because we have actual platform schematics from the platforms we were working at on the right side where you see east elevation. And so that's just so we can kind of match up where the fish is on the platforms and also to look at some of the horizontal cross beams that we'd be working with too of where they're at. And total detections on the x-axis shows our day on our left and the night on our right. And this is just the total number of detections in depth that we would get from them. And so we have that same plot when we look at our pelagic individuals, but we see a really big difference, right? So no dial differences in detection frequency. They did utilize way more of the upper portions than we see with our benthic individuals. And while we didn't get a significant difference in our dial differences, you can see a small blip during our night detections at 60 meters. So they could be utilizing the seafloor a little bit more at night, or at least some of these individuals were. And so what does this mean? Why are some at the top? Why are some at the bottom? Why is it so different by our ecotypes? And that's mainly probably because of food resources. So pelagic fishes do tend to forage more on plankton, or epipelagic fishes, more of the ones up in the water column. And benthic fishes do tend to forage on more benthic or epibenthic fauna. And when we think about epibenthic fauna, Torquato demonstrated that platforms in the Arabian Gulf have higher frequencies of functional groups actually deeper within the platforms. So all of those food sources at 35 to 60 meters for benthic individuals. So it's a great spot for them to be. And to orient you to this violin plot, we have depth, again, inverted on our y-axis, and then our x-axis, we have fish type with benthic again in that orange, and then pelagic again in that blue. And we're actually breaking it down by three different temperature bins. These are sea surface temperatures collected just from NOAA's Coral Watch, where we distinguish low as being anywhere from 22 to 25 degrees Celsius, moderate 26 to 30, and higher temperature ranges from 30 to 35. And so our benthic fish utilize deeper depths and lower sea surface temperatures, but they did display greater differences in depth distributions in moderate to high sea surface temperatures. Pelagic fish utilize shallower depths and lower sea surface temperatures, but they still have the same effects of greater differences in depth distributions from moderate to high. And that's kind of where you can see all those delineations in the violin plot. But again, this is really slow sample sizes. And so that's kind of why we see our violin plot, especially like in those low temperatures, as being kind of skinny. And so we'll kind of break it down a little bit more into the individual. And so just taking it back onto depth behavior with temperature, pelagic fishes still occupied shallow depths in these peak temperatures, where it should actually be warmer than the bottom. Some may have a wide tolerance to temperature ranges. Increases in temperature do tend to increase metabolic rate. There might potentially be enough resources closer to the top to handle this increased metabolism. But they did increase depth range, as did these benthic individuals. So we're going to look at some observations of some of this depth movement by individual over time to see maybe if we can better understand some of those movements when they're shifting. So first kind of I guess we're actually breaking it down by species, sorry. So first looking at some of our grouper individuals, which are very economically important. They showed similar depth utilization seasonally. We were able to get detections from them from the fall to the winter. And we have day and night again, total detections, except on our y-axis, we actually have total depth. Inverted again, so very similar plot to what we've seen before without the platform schematics. The greatest, oh, and then these plots are pretty cool. So we have something very similar. We have our depth inverted on our y-axis, and we do have our platform schematics again on that right side of the map. But in the background, we have our sea surface temperatures, whereas the lighter in color, the cooler in temperature it will be. As the more red the color, it'll be the higher temperature we see. And this is just zooming in at looking at some of the movements between the summer and the fall of this individual. And we do see some of the greatest range of movement and this is why I chose this time period to look at, because we actually do kind of see the fish moving in between some of those horizontal cross beams, right? Especially in that fall period, where it's really spreading out its movement. So all of those black dots are a detection with a depth associated with it at a certain time given. And so when seafloor temperature is increasing into winter, um, when we typically actually start to get less of a thermocline than we might see in the summertime, we do see individuals starting to increase their vertical range. And so I'll break that down a little bit more in this next slide. So this plot right here, so while most of our data was taken from NOAA sea surface temperature, we did originally actually start the project wanting to um, record temperature, and we did actually have some temperature recorders before the equipment failed in the beginning. And so this plot is showing temperature, date and time on our x-axis, split by seasonality. And these two lines are actually two of the original temperature recorders that we had. Some of our equipment got lost, and one of those temperature recorders actually fell on the sea floor, denoted by the red arrow. And so where we were actually able to get sea floor and sea surface temperature for a short period of time. And that kind of gave us one whole small season of what that could look like in terms of thermoclines. And so our red line is showing the temperature logger that was on the bottom of the platform, while as our blue one is showing the one that was at the sea surface. And so we can kind of see some of those big thermoclines happening within the summer into the fall. But when we were looking at that movement of that grouper in the fall, when he's actually increasing some of those movements in between those horizontal structures, was when we see that blip on this red on our bottom seafloor temperature, where we see this blip where it's actually increasing in temperature. And that's when our individuals are starting to shift. So what does that mean? So as temperature and metabolic demand increase, they may be moving for more food. So they were not, so we can kind of see that it was cooler, way cooler on the bottom than it was at the top of the sea surface. But as it starts to increase at that time, they might need more food to keep up with that metabolic demand. So what's better than one level of food? Two levels of food. So the platform structure basically creates this seafloor habitat throughout, just on a continuous level, just in this structure, right? Almost like a skyrise apartment building, um, where you could have multiple restaurants on each level. In Southeast China, they did see that that grouper species increased feeding rates and water temperatures between 29 to 34 C. And if we even go back to here, we can see that we're almost at that 29 C level. So it does check out with previous studies as well. So it might be a good reflection on that. And so when we look at our benthic mangrove red snapper, this fish exhibited movements very differently than our benthic grouper. So the snapper actually utilized the entirety of the water column. We weren't able to get any detections from the individual in the winter, but when we look at the fall, spring, and summer, we see a pretty wide movement in column depth. So when we look at that a little bit closer, looking from June to November, so a little bit more of that summer fall, we can see the individual is vertically shifting to lower depths as the temperature increases, especially in those beginning like June to August months, and then even into October, where this individual is moving in between these horizontal cross beams, still utilizing the entire column, but mainly hanging out at these certain depth areas. And we also see that the individual occupies shallower depths as sea surface temperatures begin to decline. So as it starts to get a little bit cooler, he's moving back to the top with just still a few short bounces back to the bottom. And it seems to stick specifically to the horizontal crossbeam structure. If we look at kind of most of these aggregated points, we can see that they're along the horizontal crossbeams, even as he's shifting in depth, as he's moving back and forth. So, and this individual is also likely crossing thermoclines, kind of as we saw in the map where we had almost like an 11 degree temperature difference between our surface and our seafloor. And this could be due to thermoregulatory behavior. So fish are ectotherms, and their core body temperature will fluctuate with the temperature of the water that they occupy. And this could enable the fish to actually cool down and maybe slow down their digestion. Shifting by these horizontal crossbeams could be predatory, so mangrove snappers are ambush predators, and they can continue to hunt while finding preferred temperatures in the water columns. That might be why we see a lot of fluctuations, but kind of still that segregated point at each of these depth levels. And now moving on to some of our pelagic. So if we look at our giant trevally displayed a wide range in depth in the summer, whereas he more congregated in the shallow areas in the fall and the summer, or in the fall and the winter. We did not have any detections from him in the spring. And so zooming kind of into that, again, that summer and fall area, he shifts vertically in warmer sea surface temperatures, but has less attachment to that structure, right? It's more of a slow decline than more of that branch patterned one. And the fish actually leaves in the fall to return when it gets a little bit cooler with a lot more constrained movement. And so we do see that it had similar vertical movement as our mangrove red snapper. But this individual, so giant trevallies are widely distributed across the subtropics as well as the tropics. And so this suggests that they have a higher tolerance for wide temperature ranges. So they might not be as affected as our mangrove red snapper is. So what we could be actually seeing is they could be mirroring potential prey. So as the temperature gets hotter and some of these individuals are moving to lower depths to try and cool off, they could just be following these individuals in order to hunt as well. So this behavior was observed for the giant trevally within the Great Barrier Reef, where they did see them following their prey items such as this and their movement patterns with temperature. And we also do see a benthic presence showing a decrease when the C. ignobilis left platform. So the less food sources that were on the platform could have meant that could have been why our giant trevally actually left at that time period. There wasn't enough food sources for him, so he moved on to maybe find a new food source. And then when we look at our longfin batfish, this one is really interesting. So the longfin batfish displayed really short deep dives below the thermocline. So this is again looking at that summer and fall time period with our sea surface temperatures in the background. But you can see some concentrated black dots actually for a short time period where it's concentrated in the top and then it's concentrated on the bottom. And so we didn't have any dial behavior. So I zoomed in on the specific time period, looking at just 3 days of these consistent dives, because this was almost a month that they were exhibiting these patterns. And there was no dial behaviors to it. So you can see that they just are at the top and they keep diving down and diving down and diving down and diving down. Um, so this consistent diving from surface to 60 meters happened again for like almost a month period of time. And so why are they doing that? What is the purpose of that? It is really peak temperatures during this time too. And they did kind of start to stop when our temperatures started to cool down. So this behavior is likely reflecting foraging behavior. Longfin batfish are have many different food sources. We've also seen certain behaviors like this in bluefin tuna when the surface biomass is low. So when the resources are low on the top, they might be diving down to the bottom to increase that load. And batfish do feed on a wide range of food sources. They feed on invertebrates, algae, zooplankton, um, pretty much anything under the sea. And one of their really closely related cousins is actually known to vertically forage, similar to this pattern, where they eat algae on the surface in the day and then zooplankton deeper at night. Because of the area that they're in, this could actually maybe enable them to maybe do it all the time instead of just dially like their cousins might do. But it definitely matches with some of those behaviors. And so, breaking it all down again, so overall depth utilization is most likely due to these food sources. Seasonal shifts in depth use suggest behavioral adaptations to this extreme environment. 35C is very, very, very much hotter than a typical system within the Atlantic, Indo-Pacific. And then some species of fish are adjusting movements to stay within this thermal preference to optimize their physiological performance. A lot of these behaviors, as these fish have been in this area for a very long time, these could be different ways of how they're kind of coping and able to still stay in the area. And then others may just be taking advantage of metabolic stress of smaller individuals, such as our giant trevally, who might not be as thermally stressed as the others. And so finally, jumping to our last hypothesis, that interplatform fish movement is going to occur frequently. So this is just a table of our traveled fish individuals, where different colors denote where our individual was originally tagged at which platform. We also have size associated with it, what date they were released at the platform, how many days they were detected at each of the three platforms. And we considered an individual to have traveled if it was just detected at more than one platform. We had 12 individuals from our 50 that traveled from their origin platform. Three of those 12 were considered benthic, and five were detected at all three of our monitored platform sites. I used a transition probability matrix to determine the likelihood of movement based on where it had previously been. So the likelihood it was to travel in between our three sites. So the ability to detect an individual at each site was going to be assumed equal for all fish. And what you see here on our map, so we're bringing us right back to that map. And each of our arrows, so orange is going to denote the movement from DP1, green is denoting the movement from DP3, and then blue is denoting the movement from DP2. And each of our arrows, the fatter they are, the more travel was either to or from the platform, and percentages are denoted each to look at the probability of transition between the two. And so we see that traveled individuals are more likely to spend most of their days at their origin platform, which is not too crazy. We tag them there. That is probably where they're more likely to spend most of their time anyway. But what's interesting is that DP3 and DP2 showed the highest connectivity levels, and DP1 and DP3 showed the lowest. And what we can also note there that we will talk about a little bit later too is that DP1 and DP3 are not connected by these epibenthic pipelines, while as our DP2 and our DP3 are interconnected. So that lowest connectivity between DP1 and DP3 those are the only monitor platforms that are not connected by our pipelines. And we do see the highest connectivity between DP2 and DP3. They may be using pathways to navigate north and south of in the Gulf as DP2 is going to be the furthest south, and then we have DP2 or DP4 all the way in the north, which was not monitored, as well as WP3, but they could be using these as continuous pathways. When we think about how long they took to travel, the shortest travel time we saw was from a queenfish that went from DP2 to DP3 just within 12 hours. The longest time was our giant trevally from DP3 to DP1, in which we did not get any detection in 72 days until he arrived at DP1. We also looked at the likelihood of travel with size. So similar to our linear regression, we did a binomial logistic regression here where we looked at probability of traveling on the y and then size in standard length in centimeters on the x-axis. And we do see that there is a higher likelihood of travel with increasing size of individuals. This also checks out because with our linear regression, we do see that with our pelagic individuals, and a majority of our traveled individuals were pelagic. And so this oil field has been in place since 1997. Movement is likely in response to habitat availability, environmental conditions, or resource competition, potentially utilizing structures as unique landmarks to navigate within this area, if it is a familiar place for them to navigate around. And so now we kind of bring it all together. We talked about a lot of stuff. So going back to the beginning, essentially, is residency. Going back to residency among tagfish did vary in the study. But we did have 75% of benthic fishes exhibit high to moderate residency, and we had 41% of pelagic individuals exhibit high to moderate residency. Some individuals meet essential requirements on these platform habitats, right? That high to moderate shows that they are getting what they need on these structures and they don't necessarily need to leave to go and look for more resources. We didn't see any seasonal differences in RI in our residency between some of our mature individuals, maybe reproducing within the structures, creating that productivity. And size influences of residency we had for pelagic fishes, but not for our benthic individuals. And then our platform structure overall, as well as during those extreme seasonal changes, we see differences in depth use between our benthic and our pelagic fishes, but both fish types exhibited wider ranges in vertical depth use with those increasing sea surface temperatures. Differences in this depth utilization and warmer sea surface temperatures did vary by species, but most appear correlated with these higher metabolic demands that happen within this extreme environment. And then within our connectivity too, so the travel probably due to this habitat availability, environmental conditions, resource competition, that highest level of connectivity between platforms connected by these epibenthic pipelines. Show that they probably are using them to navigate in their own habitat space within or throughout the entirety of the Gulf. More than half of them traveled, more than half of the traveled individuals were detected at all three monitored platform sites, showing that they probably do can utilize all of those platform sites in which to navigate to where they want to go. They're likely using these platforms as unique landmarks when traveling within or throughout the habitat, um, which is pretty incredible. And when we think about management, so what does this mean? Is this an essential fish habitat? Should we keep these in place? Absolutely. Full decommissioning would result in complete loss of biomass and productivity within this area. If reefed, two options that would probably help sustain the most productivity would be toppling, so placing the platform structure on its side, or even maybe partial removal. as the body of water of the Arabian Gulf is really shallow, toppling or partial removal would still keep so much of the integrity of the ecosystem that we have. Um, and Jeremy Clace has said in certain studies, based off where the fish are in utilizing the depth column, they could retain 90% of fish in a partial removal, especially when you keep the side, the top half, and place it next to the side. This could even actually expand some of the benthic habitat, increasing some of those functional groups when we place the top on the bottom too, or on the side, more of a continuous depth. It's unclear to what extent the pelagic community may be influenced, but they're not necessarily structure associated. They're more nomadic, so they might actually not really have too much of an effect anyway, kind of using that structure mainly just to hunt, right? Or look for those prey patches, not really relying on them. So removal could displace fish, driving individuals to really relocate to other nearby platforms, right? If we completely take them out, there's other platform sites within the Arabian Gulf. These fish still need this structure, and so it's just going to move them to another area. Um, but when we think about carrying capacity, density dependence, all of these things, you're just going to be reducing overall productivity and increasing competition for these resources when you're just adding a bunch of more individuals into a community instead of having all these separate communities. So, and these platforms may serve as unique landmarks for mobile species, facilitating movement across these broader areas. There's also like large migrations of whale sharks that come in and out of the Gulf. There's multiple oil platform sites within the Gulf. And so when you're leaving the Gulf and coming into the Gulf, these are kind of almost like freeway stop signs, like keep heading south. You're almost on the 405, so they could be utilizing it just like that. And so when they're gone, it might be really hard for these fish to navigate. They don't really have Google Maps like we do, and so they kind of need either the noise or some of that structure to help them navigate across this entirety of this gulf. So reefing the alkaloid platforms would also yield both ecological and economic benefits. We have some of these species, such as the grouper and all the trevallies, that could be contributing to harvestable stocks that Qatar and a lot of these other countries already probably are utilizing. And could not have done this huge project without Qatar National Research Fund, Qatar University, everybody there, everyone at University of Copenhagen, Total Energies for letting us do this research, California State University at Long Beach for taking me as a master's student and allowing me to do this, and then Southern California Tuna Club for sponsorship. But that's enough of me yapping. So does anybody have any lamps? Oh, I'm so sorry. I meant questions.
Thank you. Yeah, so if anyone just have any questions, please feel free to put it either on the chat or you could just unmute yourself and just ask the questions. Also for any of you guys that showed up late, if you guys could just put on the chat what professor you're in so I could just class you're in so I could just let them know. Um, sorry, just like your meme. Do you have any lamps? Um, so I'm just like kind of a little bit curious, because I know you were talking about like partial removal of the platforms, for the fish and stuff, right?
Yeah.
Um, but like on the schematic that you kind of show, it looks like it's cut like in half, and then like the other half just gets placed down on the benthic surface on the benthic surface, right? Um, would you think that it would be beneficial to just like instead of cutting down like a certain depth down, if they would just cut it just at the sea surface floor?
Sorry, I was trying to find you again. So you were talking about some of the partial removal.
Yeah.
Right. Okay. So I think sorry, I was like focused so hard on trying to get
Yes, that one.
space back there. Yeah. And so you're right. Like most of our benthic individuals are the ones that are highly resident too, right? So it could potentially even increase that productivity for the amount of benthic individuals that could occupy the area. Um, by having some of that, like almost taking the top and adding it to the bottom or even the top lane, you're kind of like doubling the amount of space that you have. Because, and since these platforms are pretty huge and the area is like so shallow, they would still probably even toppling or partial removal still take up to like 35 meters,
Okay.
35 to 60 meters of the column. The only thing they wouldn't have is like the shade. Um, but it doesn't really seem like with the heat, it really changes too much for the temperature between the top and the bottom anyway.
Okay. Thanks. Yeah, I was just kind of very curious. It's like, oh, like how depth would they cut it? Because like it did show that, some of the fish was still using like the high vertical areas. So I was just wondering if that would you see like less of that verticals, like them using more like the upper surface while feeding? I was just very curious about that.
Yeah, no, and so I mean from what like some of the other studies have said, like so the pelagic, since they don't like really necessarily care about the structure as much, so they would probably still be up there, kind of like above. Like sometimes even you see on seamounts, like some of those pelagic individuals will still be like high above it. Um, and yeah, and that's I guess that's kind of the tough thing too in this area since it is so shallow, because like, in California, you cut it in half and you have safe boating traffic, it's not really that big of a deal, but here it's kind of still pretty, 35 meters like low is not really that crazy. Like you couldn't put freight boats over that. So I don't know. It'd actually probably be not too much of an effect, maybe sometimes with the benthic community.
Okay. Thank you. So there is a question on the chat. They're asking, do you know if there are any data about the habitat prior to the structures? It would be interesting the before and after.
Yeah, I wish there was any data about the habitat prior to the structures. I mean, the really cool thing about the Arabian Gulf is there is some data, like historically, because artificial reefs were actually passed down um, sultan to sultan in within his family line for different like boats that would crash. But in terms of like the structures, this was even the first of its project or the first of its time to even do like a rigs to reef project in the Arabian Gulf in general. So I don't think there's much historical data actually before the platforms.
Okay. There's another question in the chat saying, you mentioned that these platforms are unique landmarks, but when whale sharks migrate, they use the noise produced to navigate. I'm just curious about what kind of noise they would hear.
Yeah, I just said noise. So it could be visual or it could be noise. As these platforms are still like they're still drilling, they make a lot of underwater noise just from either drilling or bringing the oil to the surface or even just the construction sometimes on top of the platforms. They definitely do create a decent amount of noise, even in the epibenthic structures as you're siphoning the oil from site to site as well. So there's even kind of sound in that way where it could be extremely noisy, especially with the sound traveling in the water. Um, so they could be using both visual and sound cues. I don't know exactly like what the noise or how to mimic the sound. You can almost think of like a construction site, except maybe underwater. So sometimes that can even travel a little bit further.
Um, are these platforms like constantly running like 24 hours, or is it just during the daytime?
They're not constantly running 24 hours, at least the ones that we were working on. There's one site that is like, you can live on it, you can eat on it and everything, but for the most part, the Al Khalij oil fields, you have to boat in every single day to actually go and work on the platforms. And they only do like a certain amount of platforms every day too. And some of them are really small, so they're not all going at the same time. Like it might be one to two a day, maybe working. Sometimes it might not be any of them. Um, It just kind of depends on some of the schedule. Like sometimes even the emir would come in and want to spearfish on one of the platforms, and that would get shut down for the day, and they'd have to go do another one instead. Um, so it's kind of like schedule is all over the place.
Okay. Zane has another question asking, so does that mean they or possibly other species could also hear the pipeline carrying the oil to the shoreline?
Yeah, definitely. Yeah, for sure. Probably almost all of the individuals could hear some of that working noise.
Okay. Another question that I kind of had was, Zane says, thank you for your response. Um, so like I know you mentioned earlier that you only had like a certain amount of tags on the fish, so you had like a low sample size. Was this because you were having a hard time like catching the fish, or just like you only had like a certain amount of tags, or a combination of both?
Yeah, no, so this project was extremely tricky. So we did actually originally put out 109 tags. We weren't necessarily having a problem with the tags of the fish. We actually had more of a problem with our technology. So we were only able to get out there so often. And so we had originally put out a 12 receiver array. So we had four receivers originally on each of the three platforms. And a storm wiped a bunch of them out. We weren't able to collect a ton of them. And so that was like the first year of the project. And so we had to put out a whole new array for the last year. And so we kept tagging in three different spots. So like the first time we put the receiver array out, we tagged. When we had lost all of them, we tagged, and then we put the new ones in, we tagged. And so since we had the new array out, any fish that wasn't still being detected within the new array, I kind of had to scrap. And so that's why I only had 50 individuals left for the year. So there was some that still stayed throughout the 2 year period, um, but a lot of them were lost, which was kind of a bummer. We kind of had like a smaller range of detection too for each of the sites, but that's why it kind of ended up only being those 50.
Okay, thank you. Okay, is there any other questions for Lauren? So I'm just very curious, what was like your favorite part of being able to go to Qatar and going into these platforms?
Um, let's see. Oh my gosh, it's so funny because it always kind of felt super stressful. I didn't have my advisor go with me a lot of the times, but it was really cool. Kind of I felt like I was just in like a different kind of world. Like we would fly on a helicopter sometimes to go to the offshore oil island. And it was so cool because you could see some of the whale sharks and some of the fish before you like landed on the islands and all the we got to meet so many really cool people. I think that was probably my number one favorite thing was just all the different kinds of people that I got to meet. And I made such good great friends. Because it was this I was honestly it was definitely the people. The people were really great.
Okay, thank you. Okay, are there any other questions for Lauren? Okay. Yay. Okay, well, if not, okay, Evangelios is saying it's very interesting. Thank you. Okay, well, thank you guys for showing up today to hear this amazing talk from Lauren. I know I've heard like your talk when you defended, but like hearing it all over again, I was like, oh, this is like hearing it a second time is it was like I understood it a little bit better.
Oh, thanks. Thanks, I'm glad.
So yeah, so thank you. And I hope you guys like really enjoyed it, and I hope you guys have great holidays this month. And hopefully we guys get to see you guys on our next talk.
Woohoo! Thanks, Anita.
You're welcome. Bye.
Bye.